So बड़ी साहब स्टार्ट करें और मदान साहब नमस्कार।
हाँ डॉक्टर साहब स्टार्ट करिए, डॉक्टर अग्रवाल स्टार्ट करिए।
यस यस यस।
डॉक्टर साहब राजे डॉक्टर मदान तो ज्वाइन कर लिया ना?
हाँ 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 है।
दिख नहीं रहा है।
देख रहे हैं?
दिख रहा है? हाँ हाँ देख क्योंकि यार देखना मुश्किल होता है ना। गुड मॉर्निंग सर।
Good morning.
Good morning, Dr. Madan sahab. Welcome all the participants, all the audience for this important lecture series of ICAR for a very important cause, for a very noble cause, and that is to celebrate the glorious 75 years from India's independence, Azadi Ka Amrit Mahotsav. Friends, as you know, it's a very important activity of ICAR to have the lectures of 75 eminent speakers on this platform, and we have already completed the lectures of 70 experts till date, and today is the 71st lecture. Today's speaker, I can say, a very eminent, very well-known personality in the field of veterinary science, Vigyan Ratna, Dr. Moti Lal Madangi. We welcome you, sir, and we thank you once again for accepting our request to deliver this lecture to the august audience. Our audience includes many vice chancellors, the ICAR senior officers, the senior faculty from the universities, scientists, principal scientists, head of the divisions, directors from ICAR, and to other participants. We just allow the view through the live streaming. So there are various platforms through which it is being live streamed. So we welcome you, sir, on this platform and to deliver this lecture. Friends, I'll just introduce briefly. There is a lot of contribution of Dr. Madan, but I can only say a few words because if you just see, and his contributions are well known. So that itself will take me a lot of time to elaborate his great contribution. Friends, Dr. Moti Lal Madan, Padma Shri awardee, is a distinguished veterinarian, agricultural expert, animal biotechnologist, academician, and research manager who has pursued knowledge and has contributed significantly in the areas of national agriculture development across India and different agri-eco-regions of the country. He pioneered research in animal biotechnology, and his research innovated several protocols for animal reproduction and health. Dr. Madan was born in Srinagar, Kashmir, and had his early education in Srinagar in National High School and Sri Pratap College. He obtained bachelor's and master's degrees, gold medalist in the BVSC and MSc from Punjab University and PhD from University of Missouri, Columbia, US. He has been honored with a degree of Doctor of Science in Canada by the oldest and most prestigious veterinary institution in North America, and has also earlier received DSc honorary causa at University of Agriculture and Technology, Kanpur, in Uttar Pradesh. Friends, if you see his great contributions, I can just say that in embryo transfer technology, he produced 10 embryo transfer calves from a single elite female in 1 year and holds the world record for producing the first ever buffalo IVF calf called Pratham through in vitro fertilization technology. It was a well acclaimed and well recognized contribution of Dr. Madan. He successfully initiated embryo cloning work among buffaloes for the first time in the world, which has taken India to world leadership in the buffalo cloning with birth of several cloned animals in India. Dr. Madan has served and provided science leadership in 5 different states in the country and has been vice chancellor of 2 prestigious universities, I can say. That is one at the Mathura and another at Akola. He has been Deputy Director General Animal Science in ICAR, Joint Director Research, and led the Prime Minister Mission programs on biotechnology as Project Director at NDRI. He is fellow of very prestigious six science academies, including National Academy of Sciences, National Academy of Agricultural Sciences, and National Academy of Veterinary Sciences. Dr. Madan is the recipient of many awards, I can say, and many prestigious awards. Some of them include highest national and international recognition awards like ICAR Rafi Ahmed Kidwai Award, Hariom Award, Om Prakash Basin Award for Science and Technology, Dr. B.P. Pal Award, highest recognition from National Academy, Dr. Sundaresan Award in Dairy Science, and many more to his credit. And the matter of pride for all of us is that Haryana government has bestowed the prestigious Vigyan Ratna Award for outstanding scientific contributions. Dr. Madan is a committed environmentalist and with a record of having planted 1 lakh trees in a year under Green Campaign. He is an ardent mountaineer, champion cricketer, having represented state and ICAR badminton champion, a very rare combination with a great academician, a great visionary, and having interest in the sports. Known for his varied and distinguished scientific innovation and his accomplishments in veterinary and animal biotechnology, Dr. Madan has contributed significantly to national agriculture, veterinary, animal science, research, and education. And looking at his contributions, the Government of India has awarded Dr. Madan with the highest award, I can say, for any academician, with Padma Shri in the field of science and technology 2022. Heartiest congratulations to you, and we are proud of you, sir, of your contributions. The entire ICAR family is really proud of the great contributions, the great vision which you had in your field. And we again welcome you for this. And I request Dr. B.N. Tripathi, our Deputy Director General Animal Science, to kindly chair this session. Unfortunately, our Secretary DARE and DG ICAR, Dr. Tirucham Mahapatra, is not keeping good health, so he is not able to join. And he has conveyed his best wishes for this lecture, sir. So thank you very much, sir. And with the permission of the chair, I can request you to take the floor and deliver your lecture, sir. Please. Thank you, sir.
Pleasure having me here for this lecture series, and I feel that's an honor. It's very kind of the council and yourself and the Division of Animal Sciences to have considered that some of the work which we have come out should come in a fashion that it will bring to us some future perspectives. I am also honored that this lecture is being held at a time when we are talking of 75 years of our independence. And therefore, as well, I was very young, I think a few years old, that when India got independence, but still that I think that there is a great legacy that which has gone down the road all these years, and to be continued working in this area is a great privilege that I, as a veterinarian, as an animal scientist, have been able to contribute in a small fashion to the knowledge and to the working through which that we'll be able to find that our improvement in our livestock. When I was told about this lecture, that I thought that what is the best topic I should be speaking on. And I think that Dr. Tripathi, Deputy Director General, was very explicit. He said that well, you don't need to be told about a topic, you have your own contributions, but we'd like you to spell something about the future, that what we should be doing. So I think that I will try to make a combination of this. And if I have the pleasure and the privilege to put in the slides, I think I will go on to a formal presentation. think this is.
Just click on this share screen, sir, and then share your
I am
slide.
trying to, but it is taking me. How to shift the mustn't windows. System information.
So first you open the slide, open the file, and then share.
I have them already in there. Microsoft. I don't know why. There's a little.
Uh,
Vision what should be.
सब पहले पहले अपने साथ स्लाइड पर खोल लीजिए अपना.
हाँ जी मैं तो
any problems are being faced? This?
I'm at it. I'm right at it. I hope that it should be going in the next few minutes. I opened it up.
After opening, sir, just click on share screen and select that file which is open.
नहीं वो तो open हो गई है. The presentation is right in front of me.
In share screen, sir, you will get in that window that file which is open.
Yeah,
And just select it.
Is it in there?
Not yet, sir.
ऐसा सजेशन है कि आप Alt+S दबाइए.
हमारी तो यहाँ पर आ गई है, मेरी स्क्रीन पर आ गई है.
नहीं वो शेयर नहीं हुआ सर अभी. आप पहले ग्रीन बटन दबाइए नीचे सर शेयर स्क्रीन का.
हाँ वही तो.
And then you select that file which is open, that PowerPoint. Then it will come.
I have my presentation in front of me.
Yes, now it's coming, sir.
आपने अभी कभी खोला ही नहीं है अभी.
नहीं ये तो किसी और का स्क्रीन है.
तो आप अपना दिखा रहे हैं हमें.
किसी और का प्रमोद राउत का आ रहा था किसी का. डॉक्टर साहब किसी ने सजेशन दिया कि आप लॉग आउट करिए फिर आइए आप.
क्या बात हो गई है?
और
मेरी प्रेजेंटेशन तो मेरे
सर
सामने है।
प्रेजेंटेशन आपके सामने ओपन है अभी? हेलो
हाँ
सर, प्रेजेंटेशन ओपन है आपके सामने?
हाँ,
आप विंडो का बटन दबाएंगे कीबोर्ड से नीचे सबसे लास्ट रो में थर्ड बटन कीबोर्ड पे।
कीबोर्ड पे सर एक विंडो बटन है।
हाँ, विंडो नीचे पर, हाँ, नीचे होती है, कंट्रोल के पास होती है, कंट्रोल ऑल्ट के आसपास होती है।
सर विंडो जो कीबोर्ड है उसमें विंडो बटन देखिए सर आप।
कीबोर्ड में देखिए सर,
लास्ट रो थर्ड बटन सर।
वो ऐसा वो चार रेक्टेंगल बने होते हैं ना, उस तरह का वो होता है।
चार छोटे छोटे स्क्वायर्स बने होंगे सर।
ये तो डॉक्टर साहब स्लाइड मंगा लीजिए डॉक्टर साहब से।
हाँ सर कीबोर्ड पे आपको ये विंडो बटन दिखाई देगा ना?
कौन सा विंडो बटन?
नहीं, कीबोर्ड के पे सबसे बॉटम में सर जो बटन होगा एक जिसपे चार स्क्वायर बने होंगे छोटे छोटे।
मैं तो मैं वो शेयर कर रहा हूँ।
नहीं,
आप
हाँ
खोल लेना, जो बता रहे हैं उसको
रोहित,
देखना आप।
रोहित बताओ क्या करना है, विंडो बटन से क्या करना है?
सर विंडो बटन प्रेस करके आप जूम पे क्लिक कीजिए, बीच में टास्क बार में जूम आ गया होगा। हेलो
यस,
सर, विंडो बटन प्रेस करेंगे ना तो आपको टास्क बार सबसे नीचे आएगा, वहां शो करेगा, उस जो जूम पे क्लिक कीजिए आप।
my presentation is in front of me, it is open.
Yes, it
नहीं
is in
बट
front of
इट
you.
इज
Now
नॉट
you
बीइंग
have to
शेयर्ड।
share it, sir.
लेकिन ये आप में तो क्यों नहीं जा रही है?
सर आपको ना वापस जूम पर आना पड़ेगा सर। एक बार आप मीटिंग में आइए जूम पर, प्रेजेंटेशन को हटा करके उसको क्लोज ना कीजिए, बस आप वापस से जूम पर आए।
सर क्या होता है, जैसे स्लाइड खोलते हैं पूरी स्क्रीन घेर लेता है, पता नहीं चलता कि कहाँ जाना है। तो
ये तो मैं आपके पास हूँ। I am with you, sir. I don't know what's happening here. It is shared.
सर आप मतलब आप हम सबको देख पा रहे हैं सर अभी, मतलब जूम
जी,
पर?
मैं देख रहा हूँ, सबको देख रहा हूँ। I
जी
am on the screen. I am trying to put in
सर,
this.
अब ये शेयर स्क्रीन पर आप सर क्लिक कीजिए, जो बीच में ग्रीन बटन है।
हाँ, I am sharing it.
एंड सर आपकी प्रेजेंटेशन, उसके बाद आप प्रेजेंटेशन को सिलेक्ट कर लीजिए सर।
SharePoint, optimize, Google Drive,
नहीं
Microsoft
नहीं
Drive,
नहीं
Dropbox,
नहीं, क्या आपके पास एप्पल सिस्टम है क्या सर?
share. Allow Zoom to share your screen. Open system preferences.
सर इसमें सबसे नीचे आपको आपकी प्रेजेंटेशन दिखाई, इसमें एक विंडो खुली होगी, इसमें आपकी एक प्रेजेंटेशन होगी शेयर स्क्रीन में। सर जब आपने शेयर स्क्रीन किया ग्रीन बटन, तो आपको क्या दिखाई दे रहा है सामने?
मेरी प्रेजेंटेशन में तो कुछ ऐसा लिखा नहीं है।
नहीं डॉक्टर साहब, जब आप शेयर बटन दबा रहे हैं तो क्या दिख रहा है आपको, वो पूछ रहे हैं।
स्क्रीन पर आता है वो।
नहीं, एक छोटी सी विंडो खुलती होगी ना शेयर स्क्रीन दबाते ही।
Share yours and optimize the video clip.
हाँ, इसी में सर ऊपर देखिए आपकी एक प्रेजेंटेशन खुली हुई होगी, इसमें इसमें आपका पावर पॉइंट का भी एक विंडो खुली होगी इसमें अंदर, इसी विंडो के अंदर। उस पर क्लिक करके शेयर बटन दबा दीजिए आप बस। मैं सर वीडियो कॉल करता हूँ आपको। सर ज्यादा फाइल बड़ी है, आप हमें मेल कर दीजिए सर फाइल को, वी विल शेयर फ्रॉम हियर।
डिजाइनर
सर आपके पास डॉक्टर साहब ऑफिस मत करिए, अगर आप समझ आता है तो आपको डॉक्टर डोवाल वीडियो कॉल कर रहे हैं, ठीक है, उनका वीडियो कॉल उठाइए, फिर जो वो कहें उस से करिए उस तरह से, नहीं तो इससे में यू लूज लॉट ऑफ टाइम इन दिस।
सर आप मेरा वीडियो कॉल उठाएंगे सर?
हाँ जी,
आप मेरा वीडियो कॉल उठाएंगे? मैं आपको व्हाट्सएप पे वीडियो कॉल कर रहा हूँ।
हाँ जी,
म्यूट करिए सर आप। सर आप कॉल में करता हूँ। सर मेरे को आप ईमेल कर दीजिए फाइल अपनी। आप सर फाइल ईमेल कर दीजिए मेरे को। हेलो,
क्या करूं फाइल को?
फाइल ईमेल कर सकते हैं आप मेरे को? अर्पित, अर्पित,
जी सर।
ये इनका रिमोट कंट्रोल भी तो ले सकते हैं ना हम लोग वहां से।
लीव फाइल का मैं लीव मीटिंग करूं फिर से करूं?
जी, मैं सर चैट बॉक्स में अपना ईमेल आईडी लिखता हूँ आपको।
तो ये क्या बात हो गई? Where I'm sharing it. Share queue.
नहीं, अर्पित इसमें इनका रिमोट कंट्रोल ले सकते हैं ना इनके कंप्यूटर का हम?
सर
ले तो
नहीं, वो
सकते हैं सर।
जब तक स्क्रीन शेयर नहीं करेंगे तब तक रिमोट कंट्रोल भी नहीं आएगा सर।
हेलो सर, इसमें सर एक एक करके आप फॉलो करिए स्टेप्स मैं बता रहा हूँ आपको।
हाँ बताइए।
सर ये शेयर स्क्रीन पे क्लिक करिए पहले आप।
हाँ
आपकी पावर पॉइंट ओपन है ना फाइल?
हाँ हाँ
आप शेयर स्क्रीन ग्रीन बटन जो नीचे दिया है इस पर क्लिक करिए।
हाँ ये है।
ठीक है,
Yes.
उसके बाद में एक विंडो आ रही होगी जिसमें ऊपर लिखा हुआ सेलेक्ट विंडो और एप्लीकेशन दैट यू वांट टू शेयर। उसमें सबसे पहले स्क्रीन आ रहा होगा, फिर व्हाइट बोर्ड।
नहीं यहाँ पर तो आता है ना, यहाँ लिखा है शेयर स्क्रीन।
Yes sir, please click on that.
हाँ, उस पर क्लिक कर दीजिए।
Resume stop, then there is reactions apps, and when I share screen, it doesn't go over to my.
और फिर
सर
सर जब आप स्क्रीन शेयर पर अब क्लिक करते हैं सर, तो इसमें अगर आपने एक एक कोई फाइल खोल रखी तो वही फाइल दिखेगी बस। 4 फाइल खोले तो 4 फाइल दिखेगी। तो आपने कितनी फाइल खोल रखी है?
हाँ मैं वो भी देख रहा हूँ जी, वो फाइलें नॉर्मल थी, आज पता नहीं क्या बात हो गई।
नहीं 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 साहब, आप ना डॉक्टर
आप
साहब
सर एक बार लॉग आउट करके दोबारा लॉग इन करिए सर।
हाँ
यू जस्ट लीव दिस मीटिंग एंड अगेन ज्वाइन। या आप सर ईमेल कर दीजिए फाइल को, अगर छोटी फाइल है तो। मेल कर सकते हैं सर फाइल को?
हाँ कर सकते हैं।
हाँ, मैंने अपना ईमेल आईडी लिखा है आपको भी। मेरा ईमेल आईडी है सर DDG।
आपने भेज दिया अभी?
चैट में लिखा है, अदरवाइज आप नोट कर लीजिए सर। DDG EDN शॉर्ट फॉर्म ऑफ एजुकेशन। DDGEDN@gmail.com। ओके सर, मेल। सर मेल हो गया?
हाँ
मेल कर दिया आपने ना?
नहीं मेल नहीं किया जी अभी।
ईमेल आईडी नोट कर लिया ना आपने मेरा?
Maybe I will leave the meeting then.
सर आप मेल कर दीजिए ना आप.
तो नहीं करते, मेल कर दीजिए ना, अब तक मेल कर दिए होते अब तक आप.
मेरी मुसीबत है भाई.
सर आपको कोई असिस्ट करने के लिए हो वहाँ पे तो आप उन्हें बुला लीजिए, अगर कोई असिस्ट करने के लिए है तो शायद वो आपकी हेल्प कर दें.
I will just try.
मेल तो सर हो जाएगा ना, इसलिए मेल खोलिए आप, उसमें ईमेल अटैच करके फाइल भेज दीजिए. सर आप एक्सएम्पोर्ट बोल सकते हैं, इसमें स्लाइड्स देके आप जस्ट वैसे बोल दीजिए. सर म्यूट है, आप अनम्यूट करिए. सर अनम्यूट. यू कैन स्पीक एक्सएम्पोर्ट सर, जस्ट सीइंग द स्लाइड्स.
Slide से ही होगा ये.
आपने मेल भी नहीं किया ना सर अभी.
ये मेल तो खुल गया, लेकिन ये इसके साथ अटैच नहीं हो रहा है.
नहीं, ईमेल में दिक्कत आ रही है सर को करने में.
नहीं, मैं वो उसको अटैच कर रहा हूँ जी.
अच्छा.
DDG education. DDG ASICR.
सर DDG EDN.
DDG EDN.
हाँ @gmail.com.
Gcom. Gmail. gmail.com.
हो गया सर.
हाँ जी,
DTG EDN पे किया ना,
हाँ जी,
@gmail.com.
जी.
ठीक है सर.
यूज मेल ड्रॉप पूछ रहा है ये.
साइज बड़ा होगा सर, ये हो सकता है अलाउ नहीं कर रहा हो. साइज आपकी स्लाइड्स का साइज बहुत बड़ा होगा तो अटैचमेंट नहीं हो रहा होगा.
हाँ, ये फाइल बहुत बड़ी है.
सर आप ऐसा करिए, वैसे ही बोल दीजिए सर इसको. जस्ट ओपन दिस एंड जस्ट स्पीक एक्सटेम्पो.
ये मैंने आपके पास भेजी है, फाइल आ गई क्या?
नहीं सर अभी नहीं है.
जिसने भी बनाया होगा ना लिंक,
बेटा ये स्लाइड्स नहीं चल रहा है.
स्लाइड्स का एक सेकंड.
सर कुछ हुआ क्या? ईमेल तो आपकी खुल नहीं रही है सर, जो आई है। मदान साहब, हेलो।
अनम्यूट।
ये ईमेल तो आएगी क्या आपके पास पहुंच गई?
ईमेल पहुंची है सर, लेकिन वो खुल नहीं रही है, आपका अटैचमेंट प्रॉपर नहीं है सर वो।
मैं अटैच कर दूँ।
डॉक्टर साहब इसको डॉक्टर साहब पोस्टपोन करिए। डॉक्टर साहब किस अगली मीटिंग है उसको 12 बजे, अब नहीं कर पाएंगे बैठना इसको। डॉक्टर साहब वो इनको एक आदमी बुलाना चाहिए था वहां पर, अब वो उसको अगली डेट कंफ्यूज कर देते हैं।
ओपन प्रेफरेंसेस से कोई सिस्टम प्रेफरेंसेस में
अब या तो भाई स्लाइड उनके पास है, उससे स्लाइड देख के बोल दे। तो 250 लोग जुड़े हुए, इंसान कितने लोग जुड़े हुए हैं यहाँ पर? तो चला गया था ये भेजा था हमने।
मदान साहब, हेलो सर। बेस्ट सॉल्यूशन इज आप शॉर्टली,
हाँ जी मेल में
आप स्लाइड्स देखकर सर वैसे ही बोल दीजिए एक्सटेम्पोर जो है, स्लाइड्स का चक्कर छोड़िए सर आप।
इसको मेल करनी है।
हाँ
मेल आईडी
edu gmail.com
edu नहीं सर, edn
PDN
ddg@gmail.com।
डॉक्टर साहब, डॉक्टर अग्रवाल,
हाँ
डॉक्टर साहब देखिए 11:15 बज गया, ठीक है?
सर।
अब लेटर पोस्टपोन इट, ठीक है?
अब
सर इतने सारे लोग जो जुड़े हैं, ये सर नेक्स्ट डे बड़ा मुश्किल हो जाएगा, क्योंकि एक सेकंड रुकिए सर, जरा देखता हूँ।
रखना चाहिए, किसी को बुला लेते, किसी का एनडीआरआई में पास का था उसको बुला लेते वहीं से। फोन कोई टाइम दे पाएगा। आपको डॉक्टर साहब पहले ही फाइल मंगा लिया करिए, पहले स्लाइड मंगा लिया करिए इसमें जो है।
मेल तो भेजिए आप।
सर ये फाइल खुल नहीं रही है आपकी, जो ईमेल से आ रही है अभी। सर एक ही सॉल्यूशन है, आप शॉर्टली सर वो बोल दीजिए क्या है आपका पॉइंट जो है, एक्सटेम्पोर्ट बोल के सर कर दीजिए आप उसको।
नहीं एक्सटेम्पोर का बोलना, स्लाइड डॉक्टर पास है ही, तो स्लाइड देख के अपना बोल दें अपना।
हाँ स्लाइड्स देख के तो कह रहा हूँ मैं।
हाँ
मदान साहब आप स्लाइड्स देखकर बोल दीजिए सर।
अब स्लाइड देख के बोल दो। अलाउ नहीं कर रहा है सिस्टम।
स्लाइड से करो, स्लाइड्स कहाँ गए?
हेलो
या
सर आप स्लाइड्स देखकर वैसे ही बोल दीजिए।
अच्छा
है ना? I think because people may leave now. It's already 11:50.
I don't know system.
क्या सुन ही नहीं रहे हैं वो, क्या दिक्कत हुई है? डॉक्टर मदान साहब,
कंप्यूटर हैंग हो गया, जस्ट वेट।
अरे अब उनके कोई साथ है तो डॉक्टर साहब से कहो कि स्लाइड को वहीं पर अपने स्लाइड खोल के बोल दें उसे। हम लोग बाद में स्लाइड को देख लेंगे, बाद में उसको हम अपलोड कर लेंगे, लोग अपलोड कर देते हैं स्लाइड को बाद में, सब लोग बाद में स्लाइड देख लेंगे।
अब क्या करें?
डॉक्टर साहब बोलिए, आप बोलिए अपने स्लाइड।
बोलिए सर स्लाइड।
It sounds funny, but
Sir, there is no option now.
okay. ये मैं वीडियो कैसे होगा? Am I on the video?
Yes, sir, you are on.
Yes, sir, you are on video.
Yes. Well, I think that the best way is to then start on that.
Yes, sir. Just speak, sir, as an example, by seeing your slides.
That the topic on which I will be speaking will
Yes,
be
sir.
designer technologies for productivity increase among livestock. So when we're talking of designer technologies, I'm taken back to the childhood days when we used to have a young old man coming into our house and then getting hold of the cloth and measuring it and then making clothes out of it. This situation changed subsequently over years into a shop where there will be a person standing and welcoming you and designing the clothes. And finally, these days that you don't have to go to a tailor at all, you walk into a shop. So this, in fact, is an evolution which has taken place. And this evolution has taken place right through in the process of life. And I will be talking to you about this type of an evolution, which is through my presentation, firstly, the evolution which has taken place in the formation of reproductive gonads. That is the basis of all reproduction. Then we'll be talking about what designer biology is about. Then start tinkering with the gametes, going to the designer structure and function. And finally talks about that how today we have lost our gametes and what is the epilogue out of it. Friends, when we look over into the times, we find very interestingly that if we take a picture, if we take a look of the people and the times and the animals, which as they have come up, we find that they are of all shapes and sizes. And it is something about 13.5 billion years that the whole thing started when we had the atom coming in and the physics came. Then we had the atom getting over into functional form, and we had the chemistry came in, and finally it went over into the process of biology, which was about 3.8 billion years. Since that process, things have processed over, and we have come to a stage where we have got this single cell which has formed, and this single cell has got subsequently evolved over to the animal structures, and finally an animal appeared that who was no different than other animals at that time, and it was called the sapiens. And in this process, that something very important happened. That is that the cell division which was taking place, the cell division had now two portions of it. One was the cell division, which is a simple division, and the other was the division which we call as the meiosis. And therefore, out of this meiosis division, a gamete was formed, which we call as the egg and the sperm, the two sex cells. So in the modern times, whenever we talk of reproduction, we say that our importance is for production, but no production can take place without reproduction. So as a matter of fact, that we can see this when we are talking of improvement today. So we are talking of reproduction, and we will be considering in terms of the genotypes, the phenotypes, the feed and the further efficiency of utilization. And the picture of animal improvement will not be complete unless we talk about the health, welfare, and the environment. Now, considering this, the animal improvement in this country, there are two things which become very critical. One is that we have to understand that the importance of livestock. I don't need to go over into the details of that importance of livestock, but I need to be probing into the fact that we have got a large number of our domestic livestock over in this country, and that out of them, that we have got a few of them which are foot of them which are having a definite corrector, which we give them a breed, and the rest are what we call as nondescript. And when we consider the amount of usefulness, that is amount of milk which comes out of them, we understand that it is about 78% of these animals, that which constitute about 45% of our total livestock, are the ones which are not able to produce it in terms of the good quality of milk. We get about 600 to 700 grams of them in a lactation, and the total productivity is like that. But considering all that, considering that we have got a human population of 132 million, 1,320 million, and then an animal population of 536 million, and over that a poultry population of 1,817. We consider this as a great population which is on this land mass. So taking that into consideration, that we have to see that what has been previously the method by which the animal improvement has been taking place. The animal improvement has been through the manipulation of the whole genome. We had an animal that will be see this animal has got certain genetic properties, on the basis of which we'll try to take these properties, genomic properties, and extend over into the another animal, and through what we'll be calling through a process of whole genome breeding. This whole genome breeding had for us, in terms of the improvement, a very slow process. It was slow process because I have just a message that in 2-3 minutes somebody can help me out. Just I will hold for 2-3 minutes, please.
सर क्या हुआ?
Sir, just give us 2 minutes, just 2 minutes.
सर क्या हुआ इसका, कुछ दिक्कत आई क्या?
सर वो स्क्रीन शेयर कर पाएंगे, बट ही नीड टू जॉइन द मीटिंग अगेन जस्ट वंस.
नहीं, अभी नाम तो डिस्प्ले हो रहा है इसमें,
हाँ जी, नहीं
ML
नहीं,
मदन
एक्चुअली
डिस्प्ले हो रहा है।
एक्चुअली इनके लैपटॉप में अलाउड नहीं था स्क्रीन शेयर करना, सो आई जस्ट अलाउड. ही नीड टू रीजॉइन दिस मीटिंग अगेन, देन ही विल बी एबल टू शेयर द स्क्रीन.
So he has quit.
No, he's here in the meeting right now, but he needs to quit once to share the screen. He need to rejoin. Is screen visible?
पहले वो करो, पहले स्क्रीन शेयर करो, फिर फाइल खोलो, फिर स्क्रीन शेयर करो.
नहीं, फाइल खुली हुई है, डेस्कटॉप शेयर करा है पूरा.
तो कौन सा, तो क्लिक करिए फिर उसको.
स्क्रीन विजिबल है सर आपको?
हाँ, स्क्रीन विजुअल हो है, स्क्रीन विजुअल है.
विजिबल है स्क्रीन. अब आप इसमें, आपको स्लाइड दिख रही है अभी ये लेफ्ट में यहाँ पे?
हाँ, कई सारी उसमें है, उसमें कई उसमें है, उसको आप ना उसमें ले आइए, स्लाइड शो ले आइए.
हाँ जी, हाँ जी, वो मैं कर देता हूँ अभी. मतलब स्क्रीन दिख रही है ना, वो बाकी
सब कुछ दिख रहा है, आप करो आगे, मैं इसमें
अब स्लाइड शो ले जाओ इसे. ये आप उसी बार स्लाइड शो में आओ. ये जो है ना, कब बनाया, कब पे दबाओ ये स्लाइड शो पे. अब ठीक है.
अब स्क्रीन आप इसको मिनिमाइज कर दो.
डॉक्टर अग्रवाल
जी सर.
ऐसा तो कहिए कि थोड़ा सा इसको क्विक फिनिश करने 15-20 मिनट में.
हाँ
ओके.
हाँ
हाँ जी, इसको फुल स्क्रीन.
सर, अब क्विकली थोड़ा सा लोग जाने वाले हैं, 15-20 मिनट में इसको.
Is it okay now? Or can you see?
यस सर, इट इज विजिबल सर.
हाँ जी.
हाँ, दिख रहा है सर, दिख रहा है बिल्कुल साफ.
Okay, okay. Yeah, I was.
Jump to that slide which you want.
I was just talking about these gametes. Is it okay now? Tripathi ji, are you able to see the slides?
Yes sir.
Yes, sir.
Now, I was talking to you that the critical things which came up over in the evolutionary processes is that the formation of gametes, and then this gamete by a process of meiosis separated out in the, we have then a male gamete and a female gamete. However, these differentiations become very important because we are talking in terms of having the production improvement, which will come over either through this genotype, phenotype improvements, through increasing feed and feed efficiency, or also considering the other ancillary things which come in animal improvement, that is the health, welfare, and environment. In all these fields today, the genomics and today the gene itself is playing a critical role. As I was pointing out that the productivity from our livestock, whether it is coming mostly in terms of the differentiation which is coming for the amount of milk which, for example, an indigenous cow and an exotic cow and a crossbred cow is able to produce. I think this becomes very important then when we are talking of the animal improvements and when we are talking of improving our productivity, we have to look over into the system that where we are able to get the maximum out in terms of the productivity. We consider our buffaloes, which produce about 45% of our milk. The rest is produced from the cattle, but out of them, it is the 23% of them, which you can see, that which comes from the exotic. And about 78% of the cattle do not contribute to more than 11%. So whenever we are talking of animal improvement, we have to keep into consideration this aspect, that what are the ways by which we can reach these animals and talk of improvement among the animals. Because of the fact that we have got a demand-driven economy, there is a global contribution from livestock reaching to almost 46%, and there is which exceeds the total significantly in terms of our total imports. So considering this, there is a transformation which is taking place in the agricultural system. Now, number one is that we're having much more grains available to us, which is a very good thing. At the same time, we're having milk available to us, which is now about 437 grams. But even when we consider these two things, that the food grains is available, milk protein is available, meat is available, but still we find that we're having acute shortage of nutrients. We are having malnutrition. We are having an acute impoverishment. And at the same time, that livestock, therefore, in any case, should have to play the role by which that we will be able to take the nutrition, and nutrition is here in terms of the proteins, we should reach the people, which will reach the class which is not getting at present. That in spite of the granaries being full, that we have got, in spite of the large quantity of milk being produced, that we have got mothers who are anemic, we have got the children that who die at a very early age, and we have got children who do not achieve their weight at the right moment of time. There is, I was getting into the schools lately, and I find that often that the height of the child and the age of the child do not match each other, simply because that there is acute malnutrition. So taking into these considerations, that we are talking of improving the productivity, and I was saying to it that in classical genetics, we talked about the integration of genes through a breeding of populations. And we had the advantages and certain disadvantages out of it. But we used to try to create homozygosity and from an heterozygous nature, so that we have got two genes coalescing together and making its expression feel better. For this, that there are several considerations which came. We had to take into consideration the birth weight, the reproductive cycling, the age of maturity, what is the stage of age at which the animal becomes pregnant, and then what is the mammary situation, the lactation yield, and the lifetime yield. So this used to give a data about what the animal is like, and on that basis that we'll try to use the superiority, the genetic superiority of these animals for further use. But to do that we used to take many years to find a good animal who is capable, for example, a bull, who is capable of transmitting his ability and has got a distinct higher mode of abilities. That used to take almost about 7 to 8 years. Now that's quite a time. And that's how that our progress has been very low. And at the same time, that we did not have the type of information system by which we will be able to go on to find out that where this animal had its presence felt in terms of its productivity. Now, since then, that we are now having the different natural selection of which we were talking, that the dominant factors were the environment, the fittest survival of the fittest, but there was no technology dependence. Then came that we call up the artificial selection, in which the human came in between. We started talking of a phenotype drive and then artificial selection of animals. But today, what we are talking breeding by design. In it, that we are talking of the human coming in, the technology being in there, and we are talking of the genomic additions. We are talking of the animal genome being considered for this purpose. the first headway into this animal genomic intervention came through the process of embryo transfer technology. And we had the female to be superovulated and then inseminated. And from that, instead of one egg producing at a time, that we had several eggs. And this animal was then synchronized with a female who was at the same stage of estrus, and into whom these eggs were transferred. And finally, they come out that an embryo which was there in the female reproductive tract at about 6th to 7th day of its conception was taken out and put up over into the recipient cow. And so we had this process of embryo transfer technology. I want to say this embryo transfer technology, when we started at this was in its infancy, not only here in India, but elsewhere also, as far as large animals were concerned. We especially were dealing with buffaloes here in this country. So our concern was that how to have multiple ovulations and how to use this superior animal so that we can have a faster young one being produced, so that we can have a good improvement in productivity. And this we could do, that we will be super ovulating the animal through, as you can see, the ovaries over in here. Then the zygotes will be formed. Then these zygotes at the age of about 5 to 7 days will be taken and put over in the female reproductive tract. And through that process, that we'll have the transfer processes. And once we had a very superior animal, for example, this is a 6,700 liter cow. This was one of the best animals we had here at the institute, that we got 10 calves from her in a year's time, which probably would not have been even for a lifetime to give us 10 calves, out of which about 6 calves were male and 4 were female. Now, these 6 calves, which were, we had used very high potential semen on this female, so that these young ones which were produced from them were the high potential bulls, the males from that, and the females were the one from which we had an excellent yield. And this was subsequently proved through our records, which we have. And 3 of these bulls, which produced from him, we got onto the national grid for collection and donating the semen for our reproduction purposes. But that was not that what we're all only looking for. The process of increasing the productivity was also in terms of using the material, using the genetics of this in a more faster fashion. And for that, for instance, as here you see that we had, this is from our collaborators during those places, but maybe we were on Saiwal. And there were 27 bulls produced in the country through this our program at that time, which were the elite bulls and went to different places. And from then we have got, as a matter of fact, some of the Sahiwal progenies which we have today are coming from those bulls which were at that time produced. Unfortunately, these technologies, which did not take up in the manner it should have taken off, and did not go over into our field in the manner it did. And it took almost about 15 years, or 15 to 20 years for this technology to be absorbed. And today, as I was saying it then, that you just cannot have improvement unless you use this type of a modern technology. And today, it is good that the government of India has also taken up this technology. And we have got centers all over the country now, and over 280 crores have been allotted to this, to bring about this technology so that we can have the best of the animals born, the benefits of which can go to the field, to the farmers. We had the advancement of the genetic advancement in technology is that what we have this, we had this embryo over in here, right in here, that using electromanipulation through this, an electromanipulator, that we could split this into 2. And you can have these 2 calves born and then 2 bulls produced from them. And this is what we call as the splitting identical twins which were produced. And incidentally, also these 2 Sahiwal bulls were of excellent genetic merit to be used for production purposes. The concepts came then of assisted reproduction where we have got the genome being involved. Not only is the embryo as a whole, but part of the embryo, that is the genetics. So we had the biotechnological interventions where we had the whole genome, the gene sequencing, we had then this gene tweaking, and now still further that we have got a single unit gene being worked on. So in this process, that there are a number of technologies which have come off in this animal phenomena. We have developed methods to measure existing and new phenotypes. That's what we call the state of art as it is today. We have the proteome analysis. We have metabolome analysis. We have functional genomic assays. Now, on these bases that we use gene and environment and adapting animals to, for example, the heat tolerance and producing animals which have got adaptability to the disease parameters. So herein that we have got the genome, we have got the molecular biology, we have got the bioinformatics, and we have got the functional genomics, all the three together in order that this type of advantages would be harvested. So to begin with, we had to make this young one being produced and the capability of playing with this genome. And this could be done only when we have got capabilities of producing a young one on a laboratory bench. And that's what we did at that time as an improvement of our program, is that we did this in vitro fertilization, which was done in vitro fertilization. The sperm and the eggs were on the label on the table. And with this type of an innovation, we produced these type of embryos, and then subsequently were able to produce the world's first buffalo calf from this IVF technology. That means that we are now not using the female reproductive tract, but we are taking this genetics in the form of the zygote and producing this zygote on the laboratory bench. As a matter of fact, this was at that time a big step for us here, rather for the reproductive field all over the world, that when we did over onto this. But the actual fact that which the results of it came on subsequently, when we had the excellent fashion in which that we could now reach inside the egg and inside the gene structures. So that's what that we led further over into the genomics. This is incidentally the first IVF calf born in the country, in the world. And this matured over into a good bull and proved himself to be good in all faculties, including the quality of the semen it has produced. Now, another part of this technology which came in and was very handy was that we could take these embryos and then store them. And storing them at a temperature of 196 degrees. And we have embryos stored now for maybe about 15 years, 16 years, 20 years. And they can be taken out as and when we need them and use them. This was another advantage towards the improvement, animal improvement program, so that we could have the best animal, collect as much of the gametes from him or her, and then put them over and store and produce the young ones from them. And this is what is being done at present. And this is where this activity has not only been in our laboratory, but has even traveled out of the laboratory. And that is the technologies which we call in terms of the cloning. So we can take one cell out of these, and then from one cell, we can produce a young one equivalent to that. In the process of cloning, now what we are talking of, we are taking these embryonic cells and reprogramming them. And through reprogramming an embryonic cell, we can make each cell an embryo in itself. Not only these embryonic cells, but we can also we take the epithelial cells. And that means the cell from the skin, reprogram that skin. And since we have got this technology of laboratory maturation, laboratory growth, and finding out that how these embryonic developments will take place, and then we can place it in the female reproductive tract. that we led over into the situation where we had the young ones being produced out of these cloning processes. Either through different methods for that, either through we are talking these epithelial cells and we are culturing them. Through that culturing process, we are then changing this factory, this epithelial cell. We are changing the pattern of which the division will take place, the growth will take place, the metabolism will take place. And instead of this developing over as a simple cell, that it takes up pluripotency. That means it is like a cell as it was the first cell when the development is taking place. That means when the gamete is there, when the oocyte is there, or when the sperm and oocyte meet together. So when that cell is produced, that's how we, by this process of cloning, that using an epithelial cell instead of the gametic cell. Now, this was also a great step forward in terms of the technology, that when we're now talking of taking up these epithelial cells, producing embryos out of that, and using it over into the foster mothers. In here, that thing to be noted is that we are not having any sperm. We are not having the male gamete and the female gamete. What we have got an epithelial cell, and this epithelial cell, which is a functioning cell, is then placed over into an oocyte from which the growth is taking place. And a cloned animal or a transgenic animal is produced. And we had several of these. For example, this is an animal which is born from a somatic cell. And somatic cell also not from the animals, but a somatic cell which has been collected from the semen of an animal who was already dead. So from a dead animal, semen has been stored. From that, we took a somatic cell, and that somatic cell was then processed, and the young one is produced through the process of semen cloning and embryo cloning. Not only this, but we also got over into the system, for example, a mature animal from whose cells, from an epithelial cell, from this time now, from an air cell that we cloned, and this is a young one of already a cloned animal. And from him that we got as many as 9 cloned cows. Now, these cloned cows are supposed to be genetically similar to, and there is a huge study which has been published by us, by my colleagues from ICAR, that where they have put off the abilities of these, the genetic abilities in terms of their pedigree, what they have been like. And now from the cloned bulls, which we have also seen that they are donating semen and they have got the genetic abilities. However, this must be understood that we are talking of the main gene pool is the same. But when we talk of the expression which comes from the gene tool, there are two things. One is the genes and themselves, and then the epistasis, which is coming in. And these days say that epistasis is coming up in a big fashion to find out how it influences it. And I shall be speaking about that a little later. But to take a view of this, that today, this program, national program, which we are having, that today we are having about 29 labs nationally. Where about, we have about 24,000 embryos as of the end of last month. 24,000 embryos have been collected in this IVF labs, and about 14,000 of these have been frozen because there were no ready recipients of that. About 1,446 calves have been born. And that is how this technology has been used up, and it is being used in the field and is producing its results over in the field. However, this has to be taken with caution, that when we are now talking of having these embryos being produced, the young ones born about of it, we should understand the purpose for which they are, and there should be no compromise on taking the best of the animals, because unless we are able to use the best of the animals, our subsequent productivity improvements will not be as fruitful. And similarly, we have got this through this process of IVF and taking the genes and putting it in genes, transgenics have been produced in large number of species. And here you can see the work of transgenics in animal improvement. This is the cattle in which now you don't need to dehorn them. There is a myostatin gene which has been put up, so there is more muscle into the animal. There is milk which is enriched with lysozyme lactoferrin. So the milk, it is with the animal itself, but this is done through the transgenic effects. So genetic use has been made over into the improvement of this animal. Similarly, we talk of improvement in the case of pigs, that we have got this omega-3 myostatin gene which has gone over into them. Also that there's a very important gene transgenic animal which has been produced, and that is, it is resistant to the African swine fever virus. This disease, no pig could get over into the United Kingdom. And now with the type of this transgenics which have been produced, that though United Kingdom is not very open to transgenic animals, but as far as the disease is concerned, they've accepted it. And this animal is also, for example, that the odor which used to come from the fecal matter, now that manure, that there's a low phosphorus manure, and it is because of a change which has been brought over through the genetic improvement in this animal. We are looking at the fish, for example, salmon fish, that the growth of the salmon fish has been tripled in 1 year using the growth hormone, and that Chinook salmon fishes they have gotten higher fat, they have gotten faster growing, and they are now what we call as the transgenics fish, and are greatly getting over into the grocery shelves also. Here is a picture in which that we are talking of pigs, in which the transgenes have been put up, and these pigs have now got about 7 to 8 times more growth than their litter mates, if without this gene over and being then in them. There is a suppression of one of the most dreaded diseases, avian influenza, using transgenic and genetically modified chicken have been produced, in which that this disease is preventable. Similarly, we have got what we call the stress adaptation. At one point of time, that there were about 120 eggs being produced by a hen in a year's time. And today this has gone to about 327 eggs in a year's time. And this you can see that this has put up a tremendous stress. And there is through genetics that we are talking of this gene selection and modification. To this extent, today, you can see that where we are, that in about 1957, we had an broiler rate of about 905. It went to about 78 in 1,800, and it has gone now to above weight. And you can see these using these transgenics, that it is very difficult from them even to stand. So the questions of animal welfare come in here, using these considerations. So considering these things, so there was a thought that what should we be doing about this thing? So it was that we should do that in the dairy industry to a single step, which what we call as the genomic selections. And through genomic selections is that through SNPs, through the expressions of the QTLs, that this QTL information has been collected, and on that basis, a genomic selection process has now been initiated over in different animals, including cattle. So chips have been produced, chips which have got about 50,000 SNPs, and these genomic estimated breeding values, and they compose the index with the parent averages, and then use on the basis of that chip, we can find out that what is the predict the performance, and therefore select the animals for our purposes, for our future. So what we used to do something depending upon the progeny, depending upon the pedigree. Now it is coming from the progeny. Instead of waiting for the progeny to prove itself that it is these SNPs that using these chips which are in use. That is, these are in pigs, these are in cows, these are in sheep, and in other animals. And there are various forms of this chips which are available for commercially available these days. There is also that genome editing, which is a more healthier and more fitter program. That means that where we are talking of taking a genome and editing it at a single point. That is, for example, we have got now genome modifications for increased growth, whether it is in fish or it is in salmon. There is a larger ratio of N3 to NC fatty acids, reduction in the environmental impact, avian influenza resilience, then mastitis, and resistance to the viral diseases. So particularly the viral diseases that was talking of porcine reproduction. So there are these then where we can have now the interventions, that means that the new livestock, that in them, that instead of going over into the whole gene, the advantages that we are going over into a technology which is called the CRISPR-Cas technology. As a matter of fact, that this CRISPR-Cas technology came in about 2015, but nothing was as revolutionary as when man went onto the moon and they said that a small step for man and a giant leap for mankind. I think that this has been another giant leap for mankind when we talk of this CRISPR-Cas technology system, in which we just go and touch, just as in the natural place, touch a nucleotide over in there. And by understanding that, what is the nucleotide sequence, and by understanding the structure of that gene, we can bring about a point mutation. The point mutation in itself is just like what is taking place in the nature. That means on one hand, we'll say that we are playing what nature was playing, but at the same time, we are also saying that today, now we are not talking of taking up a gene from an animal and putting in another gene, but we are only simply modifying the gene. And through that modification of the gene, we have got the zygotes and then finally the genetically engineered young one. And here we can see that we have got these chips being manufactured now in India, which is an advance which has been made. We have got one put up by National Dairy Development Board, and for cattle as well as for buffaloes. We have got one from from our institute, Biotechnology Institute in Hyderabad. As a matter of fact, they have claimed that this is the largest of the chips that they have got as a present in the world. But however, that with this indigo, as they call it, which is 11,498 this chip, this is supposed to be used, but we have these chips already in position, but we are waiting the data, evaluation data, which will come from that. What's important is that how effective we are in picking up these animals from these animals, because these chips are to be produced on the basis of the performance and the known production from the animals from which we have got the information which is going over into the NSPs. In the United States, for instance, we understand that in there are thousands of animals that who have got a very high production, and from those animals the data has been taken. So in here that we have got animal low producing animals, so it has to be seen that how validity of these chips which have been produced by us can be made. But I'm sure that in days to come that more and more advance will take place over in these areas. We have got this National Bureau of Animal Genetic Resources at NDRI, at Karnal, has also put up the chips for which they are also having a patent obtained, and they are going to be utilized that patent in the months to come in proper evaluation. we are going further over into this area of this. We are seeing that another system by which that the world over we are thinking of increasing productivity is not from animals itself, but in having the more of animal products being made. And there are two advances which have come up. One is the advances in milk, and the another is in the advances in cultured meat. Both of these are plant-derived protein sources which are available for human consumption. A cultured meat has several advantages, and similarly, that we have got people who have come out with this milk, for example, the perfect day milk, and this has come out for several reasons. One is that over the last years, having a large farm is not very economic. In 1970, there were 650,000 dairy farms, for example, in the United States of Holstein Friesians. In 2017, they got reduced about 40,000 because these farms are not very viable. And there is a great, we see that as we go over into in the years to come, that the number of farms will be going down and down. So on one hand, there is an economy in terms of economic costs of milk production are increasing. And at the same time that there are alternatives which have come up, for example, in the area of animal-free milk. And this is exactly being deciphered to be the milk as we have it. And there is properties of which have come out. And there is a great consumer acceptance for this because it's nutrients. It is comparable to the milk which is coming from the udder. So we're having this use of pluripotent stem cells, which are being put up over into the process of meat production. So instead of the cell, it is going over into the formation of a muscle, instead of it going in the formation of allowing it to go over into the system for which it has been derived at. The cell is able to multiply. A muscle cell is able to multiply and is able to produce mass. And that mass is what we call as the artificial meat these days. And this is a edible meat product and without involving animals. There are various procedures which are available for doing that process. We are also got, for example, for this, we have got the chicken meat, we have got the duck meat, we have got the beef meat, and we have got the pork meat. The pork meat that we are talking of cells, which are from the pigs, we are talking cells. So this is companies which are selling it, born out of genetics, cultured meat, for example, technologies, chicken meat, the companies, Memphis Meats, Peace Meats, then Future Meat Technologies. So there are companies who have come up for producing this meat and selling it over into the market. So this designer meat has got in the production system, production method. It is through animal farming previously. Now this is cell cultivation. So this production costs, of course, have so far very high, but if the production takes up, it will become low. We are also talking of yields. The production yields were previously low, but now they are becoming high. We are talking of characteristics, which are simple characteristics, and the sustainability is also high. Another area which is coming up is what we call as organoids. The organoids means that different organs, cells from different organs, what we say that modeling and development of biology for diseases and all that. Compared to when we talked of in vitro culture models, now we are talking of organic representative approach, where we effectively reflect the structure and function of the original organ. And these organoids are what is referred as the regenerative medicine. For example, we are talking of a regeneration of liver, regeneration of muscle, regeneration of eye. So this is what we call as organoids, organoid structure forming. And these organoid structures formings have also been now being attempted towards, for example, the udder, that we will get an udder cell being formed, so that we will have an udder, functioning an udder, not within the animal, but functioning udder outside the animal for synthetic activity to take place. And here we have got a functioning udder within the animal, and this is a biovat. This is a mammary gland which produces complex bioactive proteins that can most effectively be harvested and purified. And there is a long list of such proteins, which are of importance from health point of view for cattle, chicken, and fish, and almost all other animals. Then we also need to understand that we have been so far talking about only about the female, but there is what we talk of the bull. We used to donate the semen. As a matter of fact, semen is in real crisis now. Crisis because we need very high potential semen, and we just don't need semen because as far as reproduction is concerned, that the male gamete has been taken off from its role of having the young one being produced from it. At present, we know that males have, number of them have already reduced in the country. Valuation is through genetic potential only. And the animal energy, which used to be something to the tune of about 40% of our total agricultural energy, has come down to about 3 to 4%. And year after year, we have got about 59 to 60 lakh tractors being produced annually. And therefore, the male, even for the use of for agricultural purposes, is dwindling very fast. And then farmer feels that just for about 20 to 40 days work, that he need not idly keep an animal for about 187 or 200 days. And therefore, that the economy dictates him that he may not have a male at all. So under these circumstances, that we have to be very careful that what we do with the type of breeds which we have, and we have to nurture the best type of animals, use the females to produce the bull mothers, and from that we can go over into this functioning. But at the same time, that we have to see that how we can increase fertility. This is the most important area where the male is connected, because so far all fertilities have been projected to the female. But it is here now that we are now thinking of using fertility as an index in which that we are also going to the males. Because we have seen from the data, I was analyzing data, one of the All India coordinated projects, we found that the bulls with an fertility about 14% was also being used. Now, if the bull is a 14% fertility, we know that what can be the fertility of the herd. So therefore, that there is to be a sincere and concerned drive to find out the mechanisms by which we can increase the fertility, and we can have an estimate of the correct fertility from the males. And it is in here that we are talking of fertility sensors and marker chips. We talk of proteamine, DNA methylation, histone modification, which is taking place. And the last but not the least, the sperm sexing, which is also being started at 3 places in this country. But when we are talking of this sperm sexing, we have also been taking into consideration sex what? It's not sex we need to take a sperm, but sex, the genetics. So the important is not the sex. The important is that what genetics will be able to transfer into the females. To this extent that now the catalogs are now in preparation, and you can just dial a trait, say that in CRISPR-Cas method, that you can dial up and say that I need a trait for the young one, and that is what has revolutionized the gene editing, the new methods, that is the CRISPR craze. Also that we have got now the morphokinetic analysis being done. Today we have got possibilities of mapping every embryo not embryos as a group, but every single embryo, and taking about its through the process of algorithm, collect a large amount of data, and from that basis, subsequently, we'll have an artificial intelligence by which we can just take with a few parameters, we can find out the quality of an embryo, that how is it going. And this is getting more and more important because embryo in vitro fertilization took off from the animals, but it has proved more beneficial for human populations. Therefore, that every fertilization has now great value, and therefore they want to find out the quality of the embryo in which the semen has been used up and in which the zygote has got to develop. So to know about that, what will be the future of that zygote, that the individual embryo studies are being made. There is also designer technologies, which we talk of nano-sized multi-purpose semen sensors developed, which give us information, and then sensors about from the females, which give us information about the estrus. There are other farming technologies, automatic estrus detection technologies, for example, which have been used in grazing systems and precision livestock. And farming also needs understanding about, better understanding about the animal behavior. I will be concluding this by saying that managing reproduction and health today, that we have got the genetic control. And at one point of time, we used to go from the genome and understand the animal, that is the phenotype. But the time has come when we have now the phenotype, and we want to take this phenotype over into the genotype. Of course, that there are people who will be using this genotypic information in recreating Jurassic Park and an animal with the horns and all that. But to us, that we have to look over not to the scary part of this selection, but we have to see that how best the animal product manipulation and their physiological function for health, for resilience, and for better living, and for better productivity, we can have these animals. And I can say that for this, my message, take-home message will be that we should have a national dedicated team assigned to specific designated technologies in a mission mode with targeted output. That unless we put up a group of people together today, that by the time the other world goes ahead, that we'll be left far behind. So I am telling it, for example, CRISPR-Cas technology. For example, in genome tweeting, we should have 2 or 3 dedicated teams in this country. And then we should have redrafting of our demand scenario. Because I have seen 2 publications recently in which they feel that by 2030, we will have surplus amount of 78 million tons of surplus amount of food and surplus amount of milk and meat. But I was rather not able to appreciate that they're talking of surplus when we have got hunger and poverty in this country. That we have to take measures by which we'll be able to reach these people. And if we take into consideration the needs, the demand scenario, that we will have to relook at about the protein and energy requirements for livestock development. And to achieve envisaged targets and national initiative, I request a national initiative for national and international participation in reproductive biotechnology should make it. So I make it the name of Dare Tart, in which we say that through the designer animal technologies for reproductive technologies. There should be a unique task force of scientists, veterinary clinicians, technicians involved in the management of fertility and assessment for reproductive technologies throughout the country. And it is never too late to get started. And I hope that we'll have some thought on this issue. And it is with those words that I thank you for listening. I'm extremely sorry. I should say that there are no words. The word sorry will not be compensating the time which you people took in being patient. but I hope we got it finally, because there was a point in the computer itself that it did not log on to the type of programs it was supposed to. Before I finally say thank you to all, I would like to convey my personal, I should say, greetings and good wishes to the DGDs, to Dr. Agarwal and to Dr. Tripathi, to having stayed with me, borne me up for all this time. Thank you.
Thank you, sir. I request Dr. Tripathi to please give the concluding remarks.
I think, sir, can we have some question? I think if there is
Questions, because already we are late, so maybe
so I think so.
we will not be taking questions.
Okay. So thank you, sir, for giving a very, very exhaustive lecture. And of course, Dr. Agarwal has already given a brief biodata that we have been really regarded as the father of reproductive biotechnology in the country. And your technology that you did during 1990s is now being adopted by the government. And had it been adopted maybe around 10, 15 years back, perhaps our scenario of livestock production could have been else. And that is what countries should learn it, that how quickly we adopt our technology being developed by scientists. And that is the beauty that how we pick up and go with the parallel around the world. So today's lecture was really exhaustive, and he started with Dr. Madan has started with some basic, livestock data, human data, and human evolution. And in spite of having we have a highest livestock production, in terms of milk, meat, eggs, etc., still, our children are suffering from malnutrition, protein deficiency, and so many stunting, etc. So here is the one that we have been using traditional kind of breeding, and that takes almost 7 to 9 years when you go with an opportunity testing program. And now, so he says that production can achieve only when you go through the reproductive technology and breeding technology. They should go hand in hand, and then we can achieve our success, because our animals' productivity is very less. It is almost less than 2,000 per buffalo and maybe around 1,500 per cattle in generally. Though we have whatever milk currently we are producing, this is all the effect of technology. And the human population has increased from 1950 to 2000. Now today we have got 135 crore people. But animal population has not increased in the same pace. So therefore, whatever we are getting today in terms of milk production, meat production, egg production, they're all technology oriented. But even then, we have not been able to achieve. We have not been able to really enhance the productivity of animal. On production aspect, we are fine, but on productivity aspect, we are really poor. And therefore, such like the technology that has been started by him, starting from the embryo transfer technology, then shifting, and the first ETT calf was born, and then he started with in vitro fertilization, and where Pratham was born, and that also was a landmark achievement in India as well. But since then, and then we have gone further ahead to go with the cloning technology. We have perfected it, and he has shown that how CIRB and NDRI both are producing several cloned calf. And this is again technology of tomorrow. Today we are not adopting it. We are trying to convince the government that it should be adopted, maybe on the pilot scale, and then we can see the effect. And already CIRB and NDRI are working on that, and several cloned calves they have produced, and also they have produced their progenies as well. So this certainly will be the technology of tomorrow. Maybe after 5-6 years, we might be using the technology And then he also very clearly said that how government has adopted around 30 IVF sector has been opened in this country, almost with a cost of about 300 crore. So government is now conscious and trying to also see that our human resource development should be there, and ICAR is going and will play a big part in training the human resources here, so that these 30 centers can be really manned with a trained technician, so that IVF can become a kind of very common thing, which is now we see IVF in humans. Every city has got laboratory. So similar thing we are going to see in animals also. And when that day will come, perhaps we may have hundreds of IVF centers in the country, then perhaps our productivity and faster multiplication of our animal germplasm will be there throughout the country, and will perhaps achieve to the objective. Along with the development of new genomic tools that NDTB or NIAB Hyderabad or our NVGR, they have developed a high density chip also, and they are also combining their chips. In fact, NIAB Hyderabad chip and NVGR both are being they're working together to develop a good chip for the different trades, so that it can be easy also to adopt, and it should be cheaper also. If the cost is the price, people are not going to use that. We are also trying to work on how to these IPF, these all consumables are very expensive. So government wants that we should develop some indigenous media and consumables, so that people, it can be not very expensive to the farmers. So NDRI is also working in the direction. Then perhaps he has also gone to that many these transgenic animals, whether it in terms of disease resistant in chickens or in pigs like PRRS, and many even the Avian Influenza or other productive trait like omega fatty acids and many things, and which has really come out. The kind of technology is now there, and people are working on that. So, and also in India, also people are trying to produce transgenic animals with different traits. Our DPR, Dr. Research Hyderabad is also working on transgenic chickens with certain traits, and perhaps they will be successful in that. And Dr. Madan has also discussed about the CRISPR-Cas technology. Genome editing technology is the one that perhaps ICAR and entire NAR system should work extensively on it. Because this one technology for the production, for the health, both the technology can be used. And health also, you can use for the vaccine production, we can go for the better diagnostic development. And also similarly, it can be used in the production, in the transformation of embryo or modification of embryo, gene editing. That is all can be done by this technology. And perhaps we should work on a mission mode and should see that how this genome editing CRISPR technology is being used across our ICAR institute. Then he has also developed synthetic milk, how he developed how USA, dairy farms are decreasing, maybe up to 40,000 has come. Now they're going for the synthetic milk. Cultured milk is another area of research that researchers in India, people are working on it. Even our NRC on meat, Hyderabad, working on it. But of course, in the other parts of the world, as we stated, even chicken, duck, pork, etc., being produced as a cultured meat, and people also preferring that. Then he also talked about organoid application, that is under development, and also you have to inform that also some ICAR system and an NAR system as such organoid application for the modeling, developmental biology and diseases is also people are started working on it. And biopharma and the challenges in technology, infertility in males, they're all problems, and we are also addressing this issue by developing infertility biomarker for bulls. Normally, whichever we see that positive tested bull, we suppose that they are good, but our study says that not all bulls are fertile the way we go for using the positive tested bull. So through all these things that perhaps have been very nicely dealt with by Dr. Madan sir, and he also finally he told about the embryo pharmacokinetics, embryo health. people had gone to see that to evaluate embryo health, and perhaps his son is working in Canada on that. I had a discussion with him. So he's working on embryo health, and he developed an international laboratory as well. So similar kind of work, really, we should also take up in our institute, and perhaps that will also help us to go a step further in reproductive biotechnology. And then finally, he told about the way forward and how we can have a national task force on the program and target-oriented work in the country, so that we can really achieve our objective of getting better livestock productivity, overall livestock production in terms of meat, milk, and eggs, and also looking at our India, our country is called as a malnutrition country. It should not be like that. We should be able to see that our children are getting nutritionally sound food and our designer food, etc. So even the agricultural and crop science also, we are having biofortified crops for different kind of minerals, vitamins, etc. So if both crop sector and this animal sector both work together, then perhaps we can able to reach the objective of having a healthy India, the country. So with this, I thank you very much, Dr. Madan. Though we took a lot of time, but I knew that people is still sticking to their all their computers, and more than 150 people is still there, perhaps on physically also, and more than 230 people joined this webinar because of your reputation. You had been a great scientist, researcher, academician, administrator that we have seen. One a wonderful orator now the country can have, and one of the best scientists this country has produced in the country as far as the animal biotechnology is concerned. So with this, I thank Dr. Grawar also, and all people, all vice chancellor also, those who have joined this webinar and get benefited from the wisdom of Dr. Madan. Yes, thank you very much.
Thank you very much, Dr. Tripathi ji. And thanks once again to Dr. Madan for this excellent talk. And I thank all the audience. I see Dr. A.K. Mishra, also former chairman ASRB, and vice chancellors, many directors. So thank you very much, sir, for this excellent presentation. And I'm sure our all audience would have been greatly benefited with your great insights, great vision, which is widely acclaimed. Thank you, sir. Namaskar.
Thank you.
Namaskar, sir. Thank you.
Namaskar.